See, I'm going to discuss sex determination in Drosophila. Like humans, in Drosophila also, XY sex chromosomes are there. Males are actually XY types and females are XX types. But the mechanism of sex determination in Drosophila is very much different from humans. And let us try to understand that what are the important mechanisms of sex determination in this particular organism. See, the male Drosophila is of XY type. It possesses two sets of autosomes. That is why AA represents two sets of autosomes. One set comes from paternal side, other from the maternal side. So two sets of autosomes plus XY chromosome will be present in case of male, while in a female there would be two sets of autosomes and XX as sex chromosome. Now, males are heterogametic. It means in their case, two different types of gametes are formed. 50% of gametes will have a single set of autosome along with X chromosome, whereas other 50% will have a set of autosome and Y chromosome. Whereas in case of females, a single type of egg is formed, that is a single set of autosome plus X chromosome. So females are always homogametic. It means they produce similar type of gametes, whereas males are heterogametic because they produce two different types of gametes. So in the next generation, you can understand very well that if egg containing a set of autosome plus X chromosome gets fertilized by a sperm having autosome and X chromosome, then it will be a female individual. Okay, two sets of autosome plus XX will be maintained. And if this egg having a single set of autosome plus X chromosome is fertilized by a sperm with Y chromosome, then in that case male individual is formed. But besides this, there are other factors also, and one of the important things is the ratio of X chromosome to autosome sets. This is also important for determining sex in Drosophila. We find that if two X chromosomes are there, see the number of X chromosomes, that is two X chromosomes if present, and two sets of autosomes are there, then 2 by 2 and the ratio comes 1. If the ratio is 1, then female sex will be formed, and if this ratio is 0.5, then male will be formed. So the ratio will be 0.5 when there is a single X chromosome. So a single X chromosome and two sets of autosome will provide male. We can see this aspect in more cases, like you see here in the left side of column, chromosome formation is shown, and then ratio of X chromosome to autosomes is shown in second column, and in the third column you can see the sexual morphology. Now if two X chromosome and two sets of autosomes are there, in that situation the value, the ratio will be 1 and female sex will be formed as I just said. In case of male, if a single the ratio will be 0.5, and here in this case a single X will be there, so 1X and two sets of autosome, it will be a male. And even if Y chromosome is not there, then that individual will be a male one. Actually the presence of Y chromosome only provides fertility to the individuals who carry it. So Y chromosome is essential for maintaining fertility, otherwise maleness will be determined by the presence of X chromosome and autosome ratio, and this ratio will have to be 0.5. Now if three X chromosomes are there and two sets of autosome, then this value will be above 1, 1.5, and such individual will be a female, that is referred as meta-female. This will be very weak and will not be able to survive its full life. Likewise, if you have an individual with 3X chromosome and three sets of autosome, again the value will be 1 and this will be a normal female. Here if two X chromosomes are there and three sets of autosome, then the value will be between 1 and zero point 0.5, that is 0.67, because 2 divided by 3, the value will be 0.67, and such individual will be intersex. it will have features of male as well as female. And if a single X chromosome is there along with two autosome set, then the value will be 0.5. This will be a male. But this male will be a sterile male because Y chromosome is actually required for maintaining fertility of the individual. And if a single X chromosome is there and three sets of autosomes are there, then such individual will be meta male because the value will be below zero point 0.5 and this will be 0.33. So this type of individual will be a meta male. This will also be a very weak type of male and will not be able to survive. So you can have such individuals in which the value will be 1, that will be female. If it is 0.5, that will be a male. And if the value comes in between the two, that will be an intersex. And if it is above 1, then that will be meta female. Sometimes it is also referred as super female. And if the value comes below 0.5, that is 0.33, then such individuals will be meta male or super male. Besides this, there are certain important genes which are responsible for sex determination. In the process, we should understand that the most important gene responsible for sex determination is SXL. This SXL stands for sex lethal. This gene is found in both in both type of embryo in which a single X chromosome is there or two X chromosome is there. So in all sorts of individuals, if sex chromosomes are there and autosomes are there, then definitely this gene will be present. But what happens if the ratio is 1? If the number of X chromosome and autosome ratio comes 1 or above 1, in that situation, this sex lethal gene, it will be active. Its protein will be available in the embryo. And in such situation, the individual will develop into female. So female differentiation will occur. And if this gene is off, if its product is not available, then male individual will be formed. So important thing is that the sex lethal gene is a sexually dimorphic switch which is expressed in XX but not in X embryo. And the presence or absence of sex lethal protein modulates expression of a set of downstream genes whose products are required for sexual or sex-related variation. So this is important to understand that the sex lethal gene SXL is actually on or it is active only in those cases where the ratio is 1. It means this specific ratio is important to trigger the synthesis of this gene and to provide its product, that is sex lethal protein. So in case of female it is available, but in case of male it is not available. And its presence or absence will actually decide the other genes in the embryo to get expressed or remain dormant. So we can understand such thing in the next diagram. See, in this diagram, you can have idea that exactly what happens. The major events in the sex determination of Drosophila could be understood by seeing this diagram. See, as I just said, that in case of female, two X chromosome will be there and two sets of autosome. The ratio will be 1. In that situation, the sex lethal gene will be active and it will be translated into the protein, that is sex lethal protein. This protein is actually a splicing protein. It, along with other proteins, actually binds with a pre-mRNA, okay, hnRNA or pre-mRNA. That pre-mRNA is of transformer gene. See, this is a TRI stands for transformer. This is another gene important for sex determination in Drosophila because we should understand that there are three major genes: sex lethal, TRA, that is transformer, and the third one is double sex. DSX is for double sex. So what happens? The presence of this sex lethal protein, it actually helps the splicing of pre-mRNA of TRA. And the splicing occurs in such a manner that this TRA protein, a useful or complete form of TRA protein, is available. Actually, if you see this pre-mRNA, you find that this is initiator codon AUG. Now here UAG means it's a kind of codon which is actually terminator one. So there are introns and exon, and in the second exon, this terminator, codon is located. But what happens? The processing, actually the pre-mRNA processing occurs in such a way that second exon, which contains terminator codon, that is actually removed. So if it is removed, then in that situation, the protein TRA is formed. Okay, the TRA protein is available. And this TRA protein, along with TRA2, it helps the processing of next pre-mRNA, and that is of double sex gene. Okay, so this double sex gene, which has to provide double sex protein for female or male differentiation. So this has to be processed, and for its processing, its splicing, the TRA protein is essential. So TRA protein along with TRA2, it actually helps the splicing of the double sex pre-mRNA and helps the synthesis of double sex protein, which is female, which is female deciding actually protein. So if this female deciding protein or female specific double sex protein is available, then it binds with a protein called as intersex. IX stands for intersex, and some other proteins are also there, and it will help the differentiation of female sex. And this double sex protein, which is of female nature, it will also suppress the differentiation or development of male sex organs. Okay, male sex structures. So it functions in two ways. It actually helps the differentiation of female sex and suppresses all those genes which could help the formation of male sex. Okay, but what happens in case of male? In case of male, this sex lethal gene is not active, so its protein is not available. And when its protein is not available, then the splicing of this TRA pre-mRNA will be happening in a different way. Actually, the second exon in which UAG terminator codon is located, it is included in the mRNA. And if it is included in the mRNA, then termination occurs in between the translation process. You can say a truncated or incomplete TRA protein is available. So this is major difference. Actually, in case of female, splicing occurs in one specific manner, whereas in case of male, splicing occurs in a different way. So alternate splicing is the major happening, particularly with TRA, that is transformer gene, as well as with double sex gene. These two genes are spliced in a different manner in female as well as male, in female and male embryos. So what we are seeing here, that in case of male, the sex lethal protein is actually not available because the ratio is very less, so it is not available, and in its absence, the processing or splicing of TRA messenger RNA will be occurring in a different manner, and the protein formed, that is incomplete one, truncated one, because in the messenger RNA UAG codon is located in the middle portion of the mRNA. So protein synthesis, complete protein synthesis will not be taking place, or that kind of protein will not be available as it is available in case of female. So this is because of alternative splicing. And in the absence of TRA, the double sex pre-mRNA is spliced in a different way. And then the translated double sex protein is of male nature. That is why it is written here DSXM. That is, it is of male nature. And it will help the differentiation of male sex organs, and it will suppress the differentiation or formation of female sex structures. Okay. So in male as well and female embryo, the splicing, alternate splicing will be occurring, In female, one way of splicing will be there of these two genes, that is tra and double sex, whereas in case of other uh, male, the splicing of tra and double sex will be occurring in a different way. So this way you can have idea that there are three major players or genes which are actually responsible for male and female sex determination or sex differentiation. Now in this table you can have idea that in what way the human and Drosophila sex chromosome presence actually decide the sex. For example, in case of human we know XX chromosome if present, and here actually the autosomes consideration is not there at all. So XX chromosome that causes femaleness. In Drosophila also presence of 2X chromosome, it will cause female sex. XY they are male. In Drosophila also XY are male. But here you see XO, that is only one X chromosome in case of female. In case of female means in human if a single X chromosome is there, female is formed. But in Drosophila, a single X chromosome forms male individual. So this is major difference here. Okay, and XXX, this will be female in case of human. In Drosophila also such individual will be female. But see XXY, this will be male in case of human, and here XXY will be a female in Drosophila. So at this point you have to understand that presence of Y does not mean that it will be male in Drosophila, because Y chromosome does not play major role. And here also XXY, XXXY, this is male, but XXXY in case of Drosophila will be a female. So such type of differences are there, and I think that this much explanation will definitely help you to understand that how sex is determined in Drosophila. Although there are so many animal species and the mechanism of sex determination may be varying in different animal groups. In some cases we find that specific chromosomes are involved for sex determination, while in other cases there may be temperature as one of the major abiotic factors for sex determination. Particularly in reptiles we know the high or low temperature decides the sex. Bonellia is a marine worm in which the chemical presence, particularly on the proboscis of female, decides the sex of the individual. Those embryo which develop on the proboscis of female, they develop into male, and those which do not come in contact to proboscis develop into female. So chemicals presence will definitely be one of the major factors to decide the sex. In case of honeybees, we know that haploid and diploid set of chromosomes are responsible for sex determination. But since this mechanism has been well studied in Drosophila, its example is one of the useful examples to understand the mechanism of sex determination in this genus.